So we have the first stage, the anabolic storage pathway, and insulin driven glucose and lipid metabolism in the hepatocytes. So glycogen synthesis occurs whenever there is high glucose molecule in our blood. So whenever there is high glucose molecule in our blood, which we call what, hyperglycemia or hyperglycemia, what is happening? An enzyme called insulin is released. And when this enzyme insulin is released, it will go and phosphorylate it. So when this is happening in the muscle, it is hexokinase. It is phosphorylated. Glucose is phosphorylated by hexokinase in the muscle, but in the liver, it is phosphorylated by what? By glucokinase. Do not forget, in the muscle, we call it what? Hexokinase. In the liver, we call it what? Glucokinase. So that's all you have to know. So insulin now leads to the phosphorylation of glucose. So what is phosphorylation? Phosphorylation means addition of what? Phosphate to a glucose molecule. Addition of what? Phosphate to a substrate. So when I said I'm phosphorylating this substance, meaning that is like I'm trying to say that I am adding a phosphate group to this molecule. So example, when I said I'm phosphorylating glucose, meaning that I'm adding phosphate to glucose. So the question is, why are we phosphorylating glucose? Why can't we just go straight to the point and convert this glucose to our energy our body need? Glucose is stable. We have types of glucose. We have the open chain glucose, we have the D glucose, we have the L glucose. The D glucose is when the hydroxyl group is at the right. The L is when the hydroxyl group is at the left. So we have the open chain glucose, we have the cyclic glucose, we have the alpha glucose, and we have the beta glucose. So all these things will be treated in your carbohydrates. So we have glucose phosphorylation. We are adding phosphate to glucose because glucose is stable. Glucose is not that reactive. So for us to make it reactive, we have to add glucose. And secondly, why we are adding glucose? Because glucose is bidirectional. Look at what I mean. Glucose being bidirectional means glucose can leave the cell and move back to the blood, and it can move from the blood back to the cell. So glucose is what? Bidirectional. So it can move in both directions. But when we add phosphate, its energy is not electronegative. It is not negatively charged. So it can no more leave the cell to the blood. It will now undergo a series of processes to produce pyruvate, which now undergoes the Krebs cycle to form acetyl-CoA and now provide the energy which we need in the mitochondria. So that the process by which energy is produced in the mitochondria is the electron transport chain. We're going to discuss that when we reach about the mitochondria. So an enzyme called isomerase converts glucose 6-phosphate to what? Fructose 6-phosphate, which will now proceed the glycolysis. We are converting glucose 6-phosphate. We are converting glucose 6-phosphate to fructose 6-phosphate because we can't proceed with that fructose 6-phosphate. It is a precursor for glycolysis to occur, so we can't proceed. So the body keep on converting glucose 6-phosphate to fructose 6-phosphate. And in the formation of glycogen, what happens is what? Polymerization of phosphorylated glucose. Glucose is inactive and it is bidirectional. So when we phosphorylate it, we now make it active. We now make it negatively charged and polarized. Just this reason, it can now produce energy. So glucose, when they keep on adding, so glucose keep on adding adding. So there is a bond that exists. Have you heard of polypeptide bonds? So what are polypeptide bonds? Polypeptide bonds are bonds that joins two amino acids together. So that is also how it occurs in glucose. So glucose, we have a bond that bonds glucose together, phosphorylated glucose. They bond this glucose together. So these bonds are called alpha 1,4 glycosidic bond. Remember, in amino acid, the bond that bonds or that joins these two amino acids together are called polypeptides. Polypeptides. But in glucose, the bond that joins them together are called alpha 1,4 alpha 1,4 glycosidic bond. Glycosidic bond. So the bond that joins glucose molecule, phosphorylated glucose molecule, are called what? Alpha 1,4 glycosidic bonds. So observe something. I said glycogen is the only polysaccharide in our body, and polysaccharide are formed when monomers or when monosaccharides joins together to form straight chain, which will now branch. They keep on forming straight chain and they keep on branching. As they form straight chain, they branch. They are called polysaccharides. So that is how to identify polysaccharide: straight chain, branch, straight chain, branch. So if you observe, this glycogen is tall. There is this straight chain there, there is this straight chain, and there is these branches. See, it have go, it have branch, it have move, it have branch, it have branch. So what are you observing? So this is a straight chain, and what is happening? It have branch. What is happening? It have branch. So those parts of the straight chain are bonded by or joined together by what? Alpha 1,4 glycosidic bonds. But those branch areas, those parts where they are branching, example, look at here. Example, look at here. Here, those parts where they are branching, are bonded or joined together by what we call alpha 1,6 glycosidic bond. So glucose are polymerized by joining monomers of glucose together or monosaccharides together by alpha 1,4 glycosidic bond. But one of the characteristics of polysaccharide is the branch. So that region where the branch is joined by what? Alpha 1,6 glycosidic bond.